Today, we are looking at a breakthrough that could fundamentally change how we detect disease. We're going to explore a new way to read the hidden signals inside our blood, a technology that listens not to what our DNA says, but to how it breaks apart. Let's start with a number just to set the stage: 100 million. That is roughly how many people in the US alone are living with liver conditions. It's a huge and often an invisible public health crisis. And that's the real problem, isn't it? The damage is often silent. A lot of these conditions, like liver fibrosis, progress quietly. They can cause damage for years and years without any symptoms at all, until it's just too late for the most effective treatments. So let's dive into this silent health crisis. We need to understand why we so urgently need a better way to see what's really going on inside our bodies, long before we ever start to feel sick. Okay, so this is the whole game right here. See, liver fibrosis in its early stages is actually reversible. But, and this is a big but, if you don't catch it in time, it can lead to cirrhosis. That's permanent damage. And from there, the risk of liver cancer just skyrockets. The trouble is, the tools we have right now, standard blood tests, even fancy imaging, they're just not sensitive enough to pick it up during that critical early phase. So scientists knew there had to be clues floating around in our bloodstream. The problem wasn't a lack of information. It was more a question of perspective. Were they looking at the right signals? And that question, well, it led to a whole new way of thinking. Which brings us to our next section, where we're going to explore those very clues, the ones hidden deep inside our blood. This is the raw material that scientists are now using to find those early, whisper-quiet signs of disease. And the key ingredient here is something called cell-free DNA, or cfDNA for short. Think of these as tiny bits of DNA that are constantly being shed from cells all over your body as they die, and they just end up circulating in your blood. For a while now, we've been using this stuff in what's called liquid biopsies, mostly to hunt for specific DNA mutations linked to cancer. But here's where everything changes. This is the big idea. What if the most important message wasn't some genetic typo on the DNA fragment itself? What if the real story was in the patterns of the fragments, their size, where they came from? What if the real clue is in how our DNA shatters? This totally new focus has led to a whole new way of looking at things, what researchers are calling decoding DNA fragmentation. It's a completely different way to interpret the information that's already flowing through our veins. And this brings us to the real star of the show today, the fragmentome. We're not just talking about a few DNA fragments here. We're talking about the entire collection, a holistic genome-wide picture of the size, the distribution, and the patterns of all the cell-free DNA. It's like a snapshot of the health of your entire body. So let's break down the difference, because it's really important. The old way, the mutation-based approach, it's kind of like searching an entire library for a single typo in one book. It's super specific, super narrow. But this new method, the fragmentome approach, it's like looking at the entire library, not for typos, but for patterns. You're analyzing the size of the books, how the pages are worn, where they're placed on the shelves. You're getting a holistic picture of the health of the whole system. You know, the study's first author, Akshaya Anapragada, said it perfectly. The fact that we are not looking for individual mutations is what makes this study so powerful. It's that shift in thinking from a single target to a broad pattern-based analysis that opens the door to detecting not just cancer, but a whole range of chronic diseases. Now, as you can imagine, trying to find meaningful patterns across an entire genome, well, that's not exactly easy. The amount of data is just enormous, way too complex for a person to sort through on their own. And this is where a really powerful new tool comes into play. To give you a sense of the scale we're talking about here, the researchers analyzed around 40 million DNA fragments for every single blood sample. You multiply that across thousands of places in the genome, and you end up with a data set that is just mind-bogglingly complex. That is a job for artificial intelligence. So you might be wondering, what does this actually look like in practice? Well, the concept is surprisingly straightforward. It all starts with a simple blood draw. From that sample, the lab isolates the cfDNA and sequences it. Then, the AI gets to work, poring over those 40 million fragments, hunting for those genome-wide patterns. And finally, based on what it finds, the system can actually classify a person's risk for a specific disease, like liver fibrosis. And the results? They're pretty remarkable. When you compare this new test to the old standard for detecting advanced liver disease, it's not even close. Traditional blood markers, they catch it maybe half the time. This new AI-driven fragmentome analysis is way, way more sensitive. It finds the disease much more reliably. This kind of performance really does signal that we might be walking into a brand new era of diagnosis, one where a single blood test could give us insights into a whole host of different health conditions. And here's a really key takeaway from all this research. The platform is incredibly versatile. From that same underlying fragmentome data, you can build different, highly specific classifiers. So a test for liver fibrosis is totally separate from a test for cancer. They don't get mixed up. It's like having one master key that can be cut to fit many, many different locks. But the team didn't just stop with the liver. They went further and developed something they call a fragmentome comorbidity index. Basically, it's a score that was able to predict a person's overall risk of survival. And it actually did a better job than some traditional markers. On top of that, they started seeing these tantalizing signals, linked to all sorts of other things: cardiovascular issues, inflammation, even neurodegenerative disorders. It just hints at this huge future potential. So what's the ultimate promise here? What does this all mean? It points to a fundamental shift in how we practice medicine, a move away from being reactive, treating diseases only after symptoms show up, to being truly proactive, identifying risks and stepping in years earlier, maybe even preventing the worst outcomes before they have a chance to happen. And that leaves us with a pretty deep final thought. For almost all of human history, we've defined being healthy as simply not having symptoms. But as we get better and better at reading these hidden signals inside our bodies, you have to wonder, how are we going to redefine what it actually means to be healthy?